Here we go with part 2 on everything related to proteins, okay? Everything related to 2.4. Let's just get started. So what is denaturation? So when you learn about the topic proteins, you need to learn about a key term called denaturation. And basically what it is this. So we know that proteins, right, are made from amino acids. When you put amino acids together according to what your DNA says, you will make a polypeptide, so a chain of amino acids. We call this the primary structure. Now, this primary structure can fold into a secondary structure, okay? Either a helix like this, or a beta pleated sheet like this. Now, when these two interact with each other, this forms a tertiary structure, okay? Now, many tertiary structures can go together to form a quaternary structure, okay? And that gives us our molecule here, our basic protein, right? Now, denaturation is this. It's the process of basically undoing this, okay? It's the process of undoing this complicated tertiary structure back into a primary structure. Okay, so you don't break apart the primary structure, but you basically turn this tertiary structure back into a primary structure. And we know the tertiary structure is so important for the function of the protein, right? Let me show you here. This tertiary structure is in a specific way so that this protein can do its job. And if we destroy this tertiary structure by turning this protein back into its primary structure, by breaking all of these bonds, then it no longer has its original function, okay? So that's what denaturation is. So a structural change of a protein that results in the loss of its biological properties. So that's true. It is a structural change. We break some bonds to return it back to its primary structure, and this results in the loss of its properties, its function. So it's going to be A. Now, why not B, C, or D? So B, a change in the genetic code of an organism. No, denaturation doesn't change your original code, right? So let me show you here. You have a DNA, right? And your DNA is going to determine how these amino acids are put together into what specific proteins. Now, denaturation is the process of destroying this tertiary structure, not your DNA. Your DNA has nothing to do with this. Okay, so C, a change in the amino acid sequence of a protein, causing a disruption of its 3D shape. So the last part is true, you disrupt this 3D shape like we said, but you don't change the amino acid sequence. All you do is you return this 3D molecule back into its primary structure, but you don't rearrange these amino acids. They stay together in the same order as what they were in here, okay? So that's very important to understand. You don't change the amino acid sequence. Okay, you simply destroyed the 3D structure as I mentioned. So D, the process by which amino acids are broken down and ammonia is released. Nope, this denaturation doesn't break down amino acids, right? We know the amino acids are perfectly fine here. It's the bonding that causes them to bend and stuff to make these 3D shapes that are broken, like these bonds. So that's why the answer here is going to be A. So next one, what chemical reaction is taking place when a dipeptide becomes 2 amino acids? Okay, so they're basically saying what happens when we break apart. So here we have 2 amino acids linked together by a peptide bond. They're asking what happens when we break this bond to return these 2 amino acids linked together back into 2 separate amino acids. Okay, so we know it was called condensation to form this bond. When we have 2 amino acids, we know they react. Water is released, H2O, H2 and O are released, and we call this a condensation reaction because water is formed, okay? So this is the process of linking together 2 amino acids. So what is the process that does the reverse, okay? The process is called hydrolysis, okay? Because you use water, you put water in here to break them apart into the original thing. So it's the opposite. It's called hydrolysis. Okay, so the answer is going to be B. We know what denaturation is. Polymerization is the process of adding many amino acids together. So making a poly means many. So the action of linking together a ton of amino acids, that's called polymerization. Okay, so not D either. So it's going to be B. Now, in my teaching video, I explained that you need to know a bunch of examples of proteins, okay, including collagen, hemoglobin, myoglobin, immunoglobin, and much more. And you need to know a little bit about each of them because they can come up in questions like this one. Let me show you. So here, I'll show you here at the bottom. Okay, here are some examples. Okay, you can see there's like a list of 7-ish that you need to know. Now, this one is saying the most abundant structural protein, so one that provides strength and structure. So, for example, things that help your bones be strong, that helps your skin be strong, structural things like that, and ligaments. So what is the name of this protein? You should know that it's collagen, okay? It's going to be A. Let me show you here. Collagen here is a structural protein found in basically everything that reinforces strength, okay? It is not B because hemoglobin is a molecule inside your red blood cells that carry oxygen, okay? It's not C because this is a molecule that carries oxygen as well, just like hemoglobin. But it is specifically located in your muscles rather than your blood. Now D, immunoglobin, is a molecule that is involved in fighting sickness. Okay, it is fighting your it is involved in your immune system. So you just need to know. You don't need to know much. Just 1 or 2 sentences on what each of these are, and then 100% you can answer any multiple choice question. They're not gonna drill down and make these super complicated. They just want you to be aware of some examples of proteins. So the answer here is gonna be A. Which organic molecules may contain the element sulfur? So we know we have a whole on Earth, scientists have discovered many kinds of elements, right? That we can see on the periodic table. But maybe you haven't taken chemistry, so you're not aware of all these elements, right? Now you need to know that one of the macromolecules. Let me show you. We learned about these 4 macromolecules, like lipids, protein, carbohydrates, nucleic acids, right? Everything that we are made up, our cells is made up, is these things. Now you need to know that proteins, the one we're learning about, can have an R group, right? So we just talked about this R group. Now this R group can be any kind of molecule, And let me show you. I actually want to go back to one of the previous questions. Basically, this R group may have a sulfur in it. Okay, let me show you. This is from the part 1. So here, see, remember how we talked about in part 1 that this structure here was the linking together of multiple amino acids? And we know that this is an R group, and this is another R group, and this is another R group. Notice that some of the amino acids could have a sulfur, an S, okay? This is a sulfur as part of the R group, okay? So this is very important to know because this is useful in this question, right? We can see that, by the way, an organic molecule is any molecule that is made up of carbon, hydrogen, and oxygen, okay? So we know proteins are made up of carbon, hydrogen, and oxygen. So which organic molecule may contain this element sulfur? Proteins, okay? I just showed you the picture above. The amino acids may have sulfur in them. Not all of them, but some of them may. Whereas carbohydrates never have. Phospholipids never have. And nucleic acids don't have. So it's going to be A, okay? This is a tricky question. If you didn't know about this, that the R groups could have a sulfur in it, you may think that it's not true, but it is, okay? So I think we got one more. 1, 2, 3. Okay, we're gonna have one more for this video. So which protein has the highest tensile strength, ability to resist breaking when stretched? Okay, so this one may confuse you because you'll think, oh, it's cellulose, because that is made for structure, right? So I wouldn't blame you if you get that wrong. But the one with the highest strength, I'm not saying this is weak, this one is strong, but the one of the most strength is spider silk. Okay, it's this one here. And this is another one you need to know. So they're not just asking these out of nowhere. It is part of that little list I showed you before. See, spider silk. Okay, this is basically spider web. Okay, now basically this might be hard to believe that this is the answer, because you might think, well, spider silk's so weak I can break it. So how's it strong? The way you should look at it is like this. If you think of a steel pipe, you'd be like, wow, that's really hard. You can't break that. That's really strong. Now, if we take a zillion spider silks and put them together into the same size as a steel pipe, it will be much stronger than that steel pipe. So you shouldn't look at one single strand of spider silk, because obviously that's quite weak. But they're saying if you put them this equal size of any other material, they will be the strongest material. Okay, so this is one you kind of just have to memorize. Okay, spider silk is the answer. Actin is involved in muscle contraction. It's a protein in the muscle, and albumin is a little protein in the blood, okay, that is useful in transporting certain things around, okay? And yeah, so that's it. Let me show you here actin. We don't have actin here, but actin you'll learn about in the muscle chapter if you're going to do, I think, HL. I think it was HL. Yeah, I think it's HL. So yeah, spider silk. So that's it for this video, and again, like usual, I'll make a part 3 that will do a couple more questions. And like I said always, the more you do, the better. Your teachers is going to take most likely some of these questions, and it will appear on your test. Your teachers don't make their own questions, they use IB questions, okay? So the more you do, the better.